We are moving now to the next speaker. And this is where we're going to move into non-model species and systems ecotalks with Davide degli Esposti. I hope I didn't kill your name, Davide.
No, it's perfect, perfect.
Excellent. So if you don't mind sharing your screen and introducing briefly yourself to the
Yes,
audience, I would appreciate
of course.
this.
I have too many stuff open here, and I'm not looking. What is it? This one? No, it's not shared. Sorry. Let me close a lot of things. Okay. Yes, here it is. Sorry. Multitasking is killing part. Can you see my slides here?
Yes, we can, but if you want to go to the presenter mode, then we can
yes.
Here
Perfect.
it is. Thank you so much.
Yeah. Is it fine? Okay, great. I am Davide degli Esposti. I thank you for inviting me to this wonderful webinar and with the great presenters before me. And I am a research scientist at the National Institute of Research for Agriculture, Food and Environment in France. And my laboratory works with different model species as most of the ecotoxicology labs in the world. And I will, we are especially interested in one freshwater crustacean whose genome is still not available. And I will show you a little bit the story of the molecular approaches in these species and what we have done so far in order to understand the impact of and the mode of action of contaminants and how we can get more knowledge in molecular physiology in these wonderful organisms that help to protect and predict the effects of contaminants on biodiversity. So let's say that I will not talk again about the fact that we live in a chemical world, because I state the obvious after the presentation that we had in these 2 days. But I think that when we take the framework of the planetary boundaries, I think it's important to note that this chemical contamination that has been identified as a potential danger for human health in the 70s and for biodiversity in the decades after is actually so huge that the capacity of our societies and even the scientific communities, as we have seen, is overwhelmed in terms of evaluation. And as we have already seen by my previous colleagues, our traditional dose response toxicity testing based on laboratory acute or chronic exposures on few animal models is showing its limits, especially with the standard strategy of testing one chemical by one, and the fact that up to now we still had few crosstalks between human health and biodiversity. And as my previous colleagues also pointed out, in the last 2 decades, we had thanks to the advances in genome sequencing and nucleic acid sequencing and also mass spectrometry and the computational approaches, we have seen the development of new approach methodologies, especially in invertebrates and in model species, based on the mechanisms of action and mechanism based on the physiological knowledge and biophysical knowledge. So we have seen in silico approaches as those that Carly showed us, in vitro receptor bioassays, for example, high throughput assays, and all the omics approaches that have evolved. And all this has been framed in a context in which, as John has said yesterday, I think, we look at the conserved mechanism of toxicity across species. And this is possible in well-annotated species and model species in molecular biology and toxicology. But for what concern, oh, sorry, it exploded. For environmental relevant species, for sentinel species used in ecotoxicology, we still have few reference genomes, and this has an impact, the fact that when we perform omics approaches, we have a limited biological interpretation due to the fact that the annotations are difficult to get. So how can we develop some molecular knowledge in species that do not have still a sequenced and an annotated genome? So, so far, an approach called proteogenomics has been used in some non-model species, and this proteogenomic or more proteotranscriptomic approaches consist in coupling RNA sequencing to have a sort of molecular resources to which build a catalog of potential proteins with high-resolution mass spectrometry that validate the presence or not of these proteins. And building these protein catalogs, allows us to investigate to perform comparative proteomics studies. My PowerPoint has its own life, so it's going, okay. And this has been done by our lab in collaboration with the colleagues from the Centre of Énergie Atomique in the south of France, on our favorite crustacean, who is Gammarus fossarum. It's a freshwater crustacean, but what I am saying about our Gammarus fossarum is really transferable to many different species, and we have started to work with colleagues using decapods, so marine species, and other colleagues that are interested in mussels have asked us to contribute to their research project. So what I am telling now to you about the molecular approaches can be really extrapolated to all the species that are interested, for which there is an interest in marine sciences, for example. And Gammarus fossarum is widely used in freshwater ecotoxicologies because it is sensitive to a wide diversity of stresses. It's quite easy to identify, to sample in the field, and to manipulate. It's very frequent and abundant in Europe, and it has also an important ecological role. And we have a good knowledge of its biology. And in fact, for this species, our group and other groups have already developed a number of biomarkers, such as, for example, the measure of chemicals in the body, so measures of bioaccumulation of different contaminants, different biochemical biomarkers indicative of neurotoxicity or genotoxicity, reproduction, and different toxicological biotests based on life traits like reproduction, reprotoxicity tests, and feeding behavior, for example, up to the extrapolation of these traits to population level through modeling. So this is why we decided to invest our time in developing omics approaches and to get a more a more comprehensive approach on the molecular physiology and the mode of action of the contaminants in this species. So we'll show you some case studies in the next slides. based on basically on my work and my PhD students' work in the last few years. And the first case that I would like to show you is about reproduction. The amphipods have let's say, is a beautiful framework to work with, because their reproductive cycles, the reproductive cycles of the females in particular, is synchronized with the molting cycle, and embryonic development follow relatively independently, but the molting cycle of the female in the marsupium. So this is a perfect framework to also investigate not only reprotoxicity, but also endocrine disruption in this crustacean, due to the fact that when there is a desynchronization between the ovary maturation and the molting cycle, address the question of an effect on the endocrine system. So what I will show you here is a meta-analysis that I performed thanks to the fact that when this proteogenomic approach started in the lab, the idea was to develop the protein catalogs that were species specific, because this is the main problem when we don't have a species that is phylogenetically close. Is difficult to annotate all the proteins and the transcripts that we get. So to develop biomarkers, and the idea here was to go a little bit beyond the biomarker concept, and to anchor what the protein profiles with the phenotypes that we observe. So here what I'm showing you is a general view of the protein profiles based on a meta-analysis of shotgun proteomics data from male and female gonads and embryos at different developmental stages, with a data set that was around 400 proteins across more than 80 samples. And the idea here was to use a data-driven approach based on molecular networks to see if we were able to find protein clusters that were associated with an organ or a particular developmental stages, and if we can use this approach to discover species-specific proteins in the reproductive systems, so to improve somehow the annotation of some proteins that are unknown functions, as we often see in after downloading the annotation. So here I can show you some of the results in this case, as you on the left side there is a dendrogram, and each branch is a protein. And when we apply this biostatistical approach, this is called co-expression network analysis, that is based on R package that is called WGCNA. That I have adapted to protein counts. We saw that we were able to identify 4 different modules, and these modules were associated to different organs, the embryos, the testes, the ovaries, and in particular, what was interesting to observe was that the driving proteins associated to the embryos were involved in RNA processing and protein synthesis that were something that was expected, and also something that somehow validated the approach was the fact that we observed in the ovaries proteins that were annotated as vitellogenin, so that was what you expected. And the interesting part was that half of the protein in the ovaries were annotated as unknown proteins specific to the amphipods, close to Yalelatsek, that is another amphipod for which we have some genomic resources. And so this was a sort of case study that will help us to identify both protein clusters associated with the specific developmental stages and the possibility of annotating proteins that were previously of unknown function. And so we decided to use the same approach to explore the contaminants mode of actions. And we applied in the lab. There was a previous experiments based on the exposures of males on two insecticides, methoxyphenozide, which is an agonist of the ecdysone receptor, pyriproxyfen, that is another insecticide that is a juvenile agonist, and cadmium, who is known to be toxic for male reproduction in gammelids. So we tested 40 samples from these different cases, and from a phenotypic point of view, we observed a dose-related decrease in the number of spermatozoa in cadmium, in the red here it is indicated in red, and in pyriproxyfen treated organisms. But with the standard differential expression analysis approach, there were very few proteins that were specific to each contaminant. So we decided to see if this data-driven approach based on co-expression networks could help us to inform about the contaminants mode of actions, and here are the results. And indeed we were able to see that, and we identified in particular many different modules, groups of proteins, of which 4 were statistically associated to the different exposures, and in particular the pyriproxyfen results, where on the right side you will see a subnetwork of proteins that are associated to pyriproxyfen. And what was interesting is that, as we can see by the box plot, from a differential expression analysis, we couldn't see the association with pyriproxyfen, because also methoxyphenozide has an impact on some of these proteins. But the fact that we take into account how these proteins change their expression across the samples was able to identify this group of proteins as associated to this exposure. And when we look at the function of these proteins, we saw that these are proteins that are involved in the response of the unfolded proteins, so a sort of endoplasmic reticulum stress that in the literature seems to be a pretty conserved mode of action of testicular toxicity, and that is correlated to cell death when the stress is not resolved. So we show here that we could use this kind of approach to associate specific mode of actions to different contaminants exerting a toxic effect. And then another example that I would like to show you here is more is still data driven, but with in mind a specific task is the fact that in non-model species it's very difficult to have a coherent annotation of metabolic pathways because the tools are not really done for this. And the metabolic pathways are important to understand the contaminant impact, because they can disrupt the metabolic pathways, and we have many examples in different species on the effect of contaminants and lipid metabolism. So we decided to combine genomic tools for the annotation of genomes in arthropods in particular, and our transcriptomic data, and to include this a sort of molecular physiology approach based on proteomics to observe the specificities, the metabolic specificities of different organs. So with some colleagues that are as expert in the metabolism of insects, we I won't enter in the details, but we improved the assembly of our reference transcriptome, decreasing the impact of RNA contaminants and the redundancy that we have in the transcriptomes. And we applied a pipeline that is called CKDS that is used, that you combine different sources of annotation and can generate a coherent annotation of metabolic pathways. And the results were pretty nice because we were able to annotate more than 7,000 enzymes in our transcriptome and to cover a great number of metabolic pathways, more than 370. And most of these pathways were pretty complete. Most of them, we had a good coverage of these pathways. So we obtained for the first time a comprehensive vision of the metabolic pathways, and these resources can be used at open source, and you see here the link can be explored and navigated by everyone now. And when we go a little bit further to a more functional view, we explore the proteomic profiles in using a shotgun approach, and we focused our analysis on the proteins that were annotated in our metabolic map. So we observed here it's maybe a little bit difficult to read, but we had different organs from male and females, and the first thing that was pretty nice to observe is that globally, unless the gonads, of course, but there were not so much sexual dimorphism in the molecular physiology of the organs of the gametes. And we observed that, for example, the hepatopancreatic ceca are both a digestive organ as it is known, and a detoxifying organ, but also very active in anabolism, especially retinol metabolism or GST. And the gills are extremely active in terms of energy metabolism, of course, because this is a little bit their physiological role, and can be impacted, can be impacted by contaminants that are so I think I don't have so much time, but I would like to just to show you that from this system approach we can go back to the field and apply these approaches to develop protein biomarkers that are organ-centered in this small crustacean, using these omics data to develop the multiplexed and targeted proteomics approaches. So, and to use this using in situ active biomonitoring. So we'll go a little bit fast, but what is interesting here is, I'm sorry, I cannot, okay, is that basically this is what I would like to show you, if it works. No, okay. Based on when we look at the shotgun and targeted proteomic approach, we can cover a large diversity of annotated biological functions just with selecting 470 proteins, so that the approach is relatively unbiased. And from this, we can select the proteins that have that are specific, whose changes are specific to the different exposures. In this case, we used metals to observe these changes, and you can see the different profiles are from the different exposure conditions. And from these proteins, we can select candidate peptides that are metal specific and show those responsive behavior and are involved in the biological mechanism affected by the metal exposure, and screen these potential biomarkers in the field. Here are 92 sites were tested, and we show that most of these peptides show an upregulation or a downregulation in at least one site. So that is somehow promising to identify protein biomarkers that can be used to investigate the effect of real mixtures in these organisms. So here, to summarize, I show you that we can use a priori and data-driven systems approaches to identify molecular networks in a non-model species. And we can use this to investigate contaminants mode of action and to experiment environmental complexity. And as perspectives, we would like to integrate other omics approaches, in particular genomics and metabolomics or lipidomics, and try to build metabolic models in order to enhance the study of mode of action of contaminants, identify biomarkers of exposure or effects. And of course, comparing species through the prism of molecular pathways as Carly has shown. Okay, thank you. And open to questions.
Thank you very much, Davide.